Hello everyone. We discussed about the term genetics and also some of the important terms used in the genetics. We also discussed Mendel's experiments on garden pea and also Mendel's law of inheritance. But later on it was found that Mendel's law of inheritance is not always applicable. There is deviation from Mendel's law. Today we are going to discuss about this deviation from the Mendel's law of inheritance. First, we will discuss about modification in the monohybrid ratio. Modification of monohybrid ratio. Mendel proposed the phenotypic monohybrid ratio of 3:1. But this type of phenotypic ratio is only seen when a pair of allele has dominant and recessive relation. If no such relation is present between the pair of alleles, then this monohybrid ratio is modified. One of the example of the modification of the phenotypic monohybrid ratio is incomplete dominance. It is also known as partial dominance or semi-dominance. In this, heterozygotes express the intermediate phenotype that is not present in either of the parents or that is different from the parental phenotype. This is due to the blending of the phenotypic expression. Alleles maintain their individuality. It means these are distinct or discrete. During the gametogenesis, these segregate and then the random fusion of the gametes takes place. The offsprings that are obtained expresses their parental phenotype in the homozygous state. Heterozygotes express intermediate phenotype different from their parental phenotype. We can understand this incomplete dominance with the example of 4 o'clock plant or Mirabilis jalapa. This is the parental generation. Both are homozygous. One has red flower and one has white flower. This is crossed. Each of the plant produces one type of the gamete. These fuse and the F1 generation which we get is heterozygous and all has pink flowers. So why this is pink nor red or white? Because these two alleles do not show dominant and recessive relationship. Hence, these do not show the phenotypic expression as in the parental type. But when these are present together, this shows the intermediate phenotypic expression. And here the intermediate phenotypic expression is pink flowers. Now this F1 generation is self-fertilized. Each of the plant, male plant and female plant, produces two types of the gametes and these fuse randomly and offsprings are produced. This is the F2 generation. We can show it with the Punnett square. Here one offspring is homozygous for red, one offspring is homozygous for white and two are heterozygous. We get one red flower plant, two pink flower plant and one white flower plant. It means though in the heterozygous state, these shows the blending of the expression, but these maintain their individuality. When they are in the homozygous state, then they expresses parental phenotype. In case of capital R, red, and in case of small r, white. But when these are in the heterozygous state, then these shows the blending of the phenotypic expression, and then we get to pink flower plant. This is the phenotypic ratio. If we see the genotypic ratio, that is also one homozygous red, two heterozygous and one homozygous white. So genotypic ratio is also 1:2:1. Here the phenotypic ratio and genotypic ratio both are same. And this phenotypic ratio is not the Mendel's monohybrid ratio, but it is modified one. Second is codominance. Like incomplete dominance, in the codominance also alleles lack dominant and recessive relationship. In the heterozygous state, both the alleles express themselves. So the traits of both the alleles occur side by side, and in the F2 generation, it has both the phenotypic ratio and genotypic ratio same, that is 1:2:1. We can understand this with the example of the cattle, in which the parental generation is homozygous for the white coat color and the other is homozygous for the red coat color. Then each produces one type of the gamete, then the fusion of the gametes takes place and we get the F1 progeny, which is heterozygous. It has the roan coat color, then it is self-fertilized. Two types of the gametes are produced and the random fusion takes place between the gametes, and the F2 progeny has one white coat color, two roan coat color and one red coat color. Here both the phenotypic ratio and genotypic ratio is same, 1:2:1. Roan coat color, it is not intermediate between the white coat color and red coat color, but it is the mixture of both the red hair and the white hair. Codominance is also seen in the AB blood group and MN blood group. If the lethal alleles are present, then it results in the modification of Mendel's monohybrid phenotypic ratio, which is 3:1. What are lethal alleles? Alleles whose expression results in the death of an organism is known as lethal allele. Suppose there is certain gene present which is essential for the survival of an organism. It undergoes mutation and due to this it creates certain allele whose expression results in the death of the organism or causes the death. Then it is known as lethal allele. Lethal allele is of two types, recessive lethal allele and dominant lethal allele. Recessive lethal allele shows its lethality or causes the death of an organism only in the homozygous state. But dominant lethal allele shows lethality even in the heterozygous condition. So it is rare, because if this dominant lethal allele is present, then the individual will die before producing its progeny. So this dominant lethal allele is removed from the population in the same generation in which it occurred. Now let us see this recessive lethal allele with the example of snapdragon. In the snapdragon, three types of the plants occur. Green plant, which is homozygous dominant, and it is green in color because chlorophyll is present. Aurea, which is yellowish green, which is heterozygous, and it is yellowish green due to the presence of the carotenoids. White plant, which is homozygous for the lethal allele small c. Here chlorophyll is absent, so it dies. Two aurea plants are crossed. Each produces two types of the gametes, then these fuse randomly, and then we get the offspring in which one is green, 2 aurea and 1 albino. So we get the plants in the ratio 1 green is to 2 aurea is to 1 albino. But this albino plant has the homozygous recessive allele. It is white in color because the chlorophyll is not produced, so it dies. So the phenotypic ratio which we get is 1:2 instead of Mendel's monohybrid phenotypic ratio 3:1. In humans, the recessive lethal allele is seen in sickle cell anemia. In this, the red blood cell is sickle shaped instead of biconcave oval shaped. This affects the oxygen carrying capacity of the red blood cells and also hinders the blood circulation, which ultimately results in the death of the individual. This is the allele for the sickle cell. When it is in the homozygous condition, it results in the sickle cell anemia and the individual dies. In the heterozygous condition, where one is the normal allele and one is the allele for the sickle cell, then the individual do not exhibit or do not show any symptoms. It is indistinguishable from the normal individual, but it is carrier. It carries the allele for the sickle cell from one generation to another generation. If the individual is homozygous for the allele for normal RBC, then the individual is normal. It neither shows any symptoms nor it is carrier. This is the parental generation. 2 heterozygous individuals are crossed. Each produces 2 types of the gametes. Then their random fusion takes place and the offspring which we get is 1 normal, 2 carrier and 1 has the sickle cell anemia. So this individual dies. Hence the phenotypic ratio is 1:2 and this is also the genotypic ratio. There are 4 categories of lethal alleles. One is the early onset gene or alleles which results in the early death of the organism, mainly during the embryogenesis, is known as early onset. Late onset alleles which show delayed effect, as a result of which the individual survives for some time and later on it dies or succumb to death or death occurs, is known as late onset. Conditional lethal allele which results in the death of organism under certain environmental condition but not in the other condition, is known as conditional lethal allele. Example, temperature sensitive lethal allele which causes the death of the organism at high temperature, but the organism survive or the individual survive in the optimum temperature. Semi-lethal allele which kills or cause the death of only few individuals in the population but not all, is known as semi-lethal allele. We are going to discuss about the modification of the dihybrid ratio. Mendel's dihybrid ratio is 9:3:3:1. But in many cases, it is seen that there is the deviation from this dihybrid ratio or there is the modification in this dihybrid ratio. And this is possible due to the intragenic or intra-allelic interaction. This intra-allelic or intragenic interaction is known as gene interaction. What is gene interaction? Earlier, according to Mendel, gene function independently and one gene is responsible for a single trait. But later it was found that many different genes together contribute for a single trait in an organism. When two or more genes that are present on the different locus, that is, they are non-alleles, are responsible for a single trait in an organism, then it is known as gene interaction. One such gene interaction is seen in epistasis. In this video, we will discuss about two types of epistasis: dominant epistasis, recessive epistasis, or supplementary gene epistasis. In this, one gene masks the expression of the other gene present on the different locus. Gene which masks the expression of the other gene is known as the epistatic gene, and gene whose expression is masked is known as the hypostatic gene. But this type of the interaction is different from the dominant recessive interaction, because in the epistasis the interaction occurs between the non-alleles, that is the alleles of the genes that are present on the different locus. But dominant recessive interaction occurs between the alleles of a gene. There are different types of epistasis in which different dihybrid ratio is seen. We will discuss these epistasis one by one. First is dominant epistasis. Here the dihybrid ratio is 12:3:1. In the dominant epistasis, dominant gene masks the expression of the other dominant gene present on the different locus. Suppose capital P is the dominant allele which masks the expression of the another gene, capital R. So here P will express itself phenotypically, but it will suppress or mask the expression of the capital R. So P is epistatic to R, or capital P is the epistatic gene and capital R is the hypostatic gene. Capital P gene is responsible for the production of purple color, and capital R gene is responsible for the production of the red color. So let us understand this dominant epistasis with the example. Parental generation is crossed. One is homozygous for the dominant P and recessive R. The other is homozygous for recessive P and dominant R. So it has the purple flower color and it has the red flower color. Then these produces gametes, which fuses and produces the F1 generation, which has all purple flower. Here, if we see the genotype, it is heterozygous for the P allele and also heterozygous for the R allele. So why it is purple? Because this dominant P is epistatic to dominant R. So this dominant P or the capital P masks the expression of the capital R. So the red color is not produced and the dominant P expresses itself phenotypically. Hence all the F1 generation is purple. Then this F1 generation is self-fertilized. All the male and the female plants produces 4 types of the gametes each, and we get the offspring in which 12 are purple, 3 is red and 1 is white. So this 12:3:1 is the phenotypic ratio. Now let us discuss about this ratio, or let us see the explanation of this ratio. When the dominant allele P is present, then it codes for the functional enzyme P. In the presence of the dominant allele R, it encodes for the functional enzyme R. Both the enzyme works on the white substrate. Enzyme P converts the white substrate into purple product, and enzyme R converts this white substrate into the red product. In the genotypes in which both the dominant allele P and dominant allele R is present, we get purple color flowers. Like in the F1 generation, all flowers are purple. Why we get purple flower and not the red flowers? This is because the enzyme P and R competes for the white substrate, but the enzyme P has greater affinity for this white substrate compared to the enzyme R. So if both the enzyme P and the enzyme R are present, then this enzyme P will convert all the white substrate into purple product, and hence we get all purple flowers. So if we see these genotypes, at least one dominant allele P is present. Hence functional enzyme P is present and white substrate is converted into purple product. So we get all the plants having the purple flowers, and the number is 12. If we see these genotypes, here the dominant allele P is absent, or there is the lack of the dominant allele P. In the absence of the dominant allele P, enzyme P is not functional, but here at least one dominant allele R is present. So in both the cases, enzyme R is functional. In the absence of this enzyme P, now enzyme R works effectively on this white substrate and converts it into the red product. So in this case, the plant that we get has the red flowers, and here the number is 3. If we see this genotype, it has both homozygous recessive P and homozygous recessive R. So here both the dominant P and dominant R is absent. So there is no functional enzyme P or functional enzyme R. Hence this white substrate is not converted into any color product. So the plant that we get has white flowers. And here the number is 1. So in this way we get 12 purple, 3 red and 1 white flower color plant. So the phenotypic ratio is 12:3:1. Now recessive epistasis or supplementary genes. When recessive alleles present on one gene locus in the homozygous condition mask the expression of non-alleles, that is genes which are present on the another locus, then it is known as the recessive epistasis. So the hypostatic gene expresses itself only when the epistatic gene is dominant, either in the homozygous or the heterozygous state. So let us understand this. This is the parental generation. One red flower plant is crossed with the white flower plant. Here the red flower plant has homozygous dominant R and homozygous recessive P. White flower plant has homozygous recessive R and homozygous dominant P. F1 generation which we get is all purple. Then this F1 generation is self-fertilized and we get F2 generation. This F2 generation has the ratio 9 purple flower plant is to 3 red flower plant and is to 4 white flower plant. So how it is possible? White substance which is present acts as the substrate for the enzyme R. This enzyme R is encoded by the dominant R gene. This then acts on the or works on the white substance and converts it into the red product. This red product, in the presence of the functional enzyme P encoded by dominant P gene, is converted into the purple product. So here we can say that this dominant R is independent. That means it expresses itself, it shows its phenotypic expression, that is the red color, either in the presence of the dominant P or in the absence of this dominant P. But P, it produces its effect, or it expresses itself, or it expresses the purple color in the presence of the dominant R. Because when enzyme R converts the white substance into red product, then this red product acts as a substrate for the functional enzyme P, and this enzyme P then converts this red product into purple product. Now let us see the genotypes of the F2 generation. Here the genotypes which has at least one dominant R and one dominant P gives the purple flower color, because here both the functional enzyme R and functional enzyme P is present. So the white substance is ultimately converted into the purple product. So we get 9 purple flower plant. In this type of the genotype, where at least one copy of dominant R is present, but homozygous recessive P is present. So only functional enzyme R is present, but there is the absence of functional enzyme P. So this white substance is converted into the red product, but it is not further converted into purple product in the absence of functional enzyme P. So in this type of the genotypes, we get red flower plant and its number is 3. Now in the third type of the genotype, where the homozygous recessive R is present, or at least one copy of the dominant P is present. So here this enzyme R is non-functional, but enzyme P is functional. In the absence of this functional enzyme R, the white substance is not converted into red product. So if the red product is not present, then there is no substrate for the functional enzyme P. So the purple product or the purple color is not produced, as a result of which plant has white flower instead of the purple flower. In the fourth type of the genotype, both the R is in homozygous recessive condition and P is also in the homozygous recessive condition. So there is no functional enzyme R or P. Hence, no pigment is produced or white substance is not converted into any color. Hence, the plant gives white flower instead of red or purple flower. So if we see the total, we get white flower plant, that is 4 in number, and this explains the F2 ratio 9 purple is to 3 red is to 4 white. If we see this genotype, here gene R is present in recessive homozygous condition and gene P has at least one dominant allele, and the plant we get has white flower color, because in the presence of the recessive homozygous R gene, enzyme R is not present, so white substrate is not converted into red product. So even in the presence of enzyme P, purple product is not formed in the absence of this red product. So in this way the expression of dominant P allele is masked, and this is masked due to the presence of this homozygous recessive R allele. So recessive R gene is known as the recessive epistatic gene. Also, dominant R gene expresses itself independently, but dominant P gene does not express itself independently. It requires the presence of the R gene for its phenotypic expression, that is the purple color. Hence, R and P genes are known as supplementary genes. Now let us see the comparison between dominance and epistasis. Dominance is allelic suppression. Epistasis is non-allelic suppression. Dominance involves single pair of alleles. Epistasis involves two pair of alleles. In dominance, gene suppresses expression of its allele. In epistasis, gene suppresses expression of non-alleles. In dominance, effect of recessive allele is suppressed. In epistasis, effect of dominant or recessive allele is suppressed. Dominance is due to dominant allele. This dominant allele masks the expression of recessive allele. Epistasis is due to dominant and recessive allele. We are going to discuss about duplicate gene and modification of Mendel's dihybrid ratio. Now let us see duplicate genes with cumulative effect. If the dominant alleles present at the two different gene locus are responsible for the phenotypic expression of any trait together, and if these dominant alleles present on the either of the gene locus results in the same phenotypic expression individually, then these are known as the duplicate genes with cumulative effect. One of the example is the coat color in pigs. The parent and generation both having the sandy coat color is crossed. One has the dominant R allele in the homozygous condition and recessive S allele in the homozygous condition, and the other has recessive R allele in the homozygous condition and dominant S allele in the homozygous condition. So each of them produces one type of the gametes. Then these fuse and the F1 generation is all red. Then this F1 generation is self-fertilized and we get the offsprings, 9 having the red coat color, 6 having the sandy coat color and 1 having the white coat color. So the phenotypic ratio here is 9:6:1. So let us see the genotypes of these F2 offspring. Here in the first type, where the coat color is red, at least one dominant R and dominant S is present. So dominant R and dominant S is present together, either in the homozygous condition or heterozygous condition. In second type, which has the sandy coat color, either dominant R allele is present or dominant S allele is present in homozygous or heterozygous condition. In third type, both the R allele and S allele is present in the recessive homozygous condition. So from here we can conclude that when both the dominant R allele and the dominant S allele is present, either in the homozygous condition or heterozygous condition, these together produce the red coat color, or these together is responsible for the red coat color of the offspring. But if dominant R allele or dominant S allele is present, either dominant R allele is present or dominant S allele is present in the homozygous or heterozygous condition, then the offspring has sandy coat color. So from here we can conclude that this dominant R or dominant S, when present in the homozygous or heterozygous condition, these individually produces the same type of the phenotype. Hence R and S is known as duplicate genes. And when these are present together, then they produces the red coat color. So these show the cumulative effect when present together. Hence the dominant R and dominant S allele that are present in the two different gene locus are known as the duplicate genes with cumulative effect. When the recessive gene in the homozygous condition present at the two different gene locus produces the same or identical phenotypic expression, then these are known as the duplicate recessive gene. When the dominant allele of two gene locus are present together and they complement each other and results in the different phenotypic expression, then these are known as the complementary genes. Now let us understand this with the example. Two parental generation is cross, both has the white flower color. One has the homozygous recessive A gene and homozygous dominant B gene, other has homozygous dominant A gene and homozygous recessive B gene. F1 generation is heterozygous for both the genes and these are all red. F2 generation has 9 red flower plant and 7 white flower plant. So the phenotypic ratio is 9:7. Now let us understand this 9:7 ratio with the genotype of the F2 generation. In these type of the genotypes, at least one dominant A allele and one dominant B allele is present together. These complement each other and produces red phenotype, which is different from the white phenotype. So how this red color is produced? Dominant gene A is responsible for the chromogen. Dominant gene B encodes for the functional enzyme B, which works on this chromogen and produces the colored product red. So here both dominant gene A and dominant gene B complement each other and produces the red color. In this type of the genotype, at least one gene has the recessive allele in the homozygous condition, and these produces the identical phenotype, which is white flower color. This homozygous recessive allele A and homozygous recessive allele B, these are present on the different gene locus, but these produces the same type of the phenotype. Therefore, homozygous recessive allele of A gene and homozygous recessive allele of B gene produces the same type of the phenotype. So these are known as the duplicate recessive gene. So the dominant allele A and dominant allele B, when they are present together, they complement each other and results in the phenotypic expression red color, which is different from the white color. So these are known as the complementary genes. Now let us see duplicate dominant interaction where the dihybrid ratio is 15:1. When dominant alleles of two gene loci produces same phenotype or same phenotypic expression, and when dominant alleles of two gene loci present together do not show any cumulative effect, then it is known as the duplicate dominant interaction. One of the example is seed capsule of shepherd's purse, scientific name Capsula bursa pastoris. It has two types of seed capsules, one is triangular shaped and the other is the top shaped. In this, the parental generation, triangular and top shaped is cross. Triangular is homozygous dominant for both the genes and top shaped is homozygous recessive for both the genes. F1 generation we get is heterozygous for both the genes and all are triangular in shape. F2 generation has 15 triangular and 1 top shaped. Ratio is 15:1. Now let us see the genotype of this F2 generation. In this type of the genotype, at least one dominant allele of both the genes are present together. In this type, either dominant allele of gene A is present in homozygous or heterozygous condition, or dominant allele of gene B is present in the homozygous or heterozygous condition. So all these genotypes give triangular seed capsule. Number is 15. So how all these genotypes are giving the same type of the seed capsule, triangular. Dominant gene A encodes for enzyme A, dominant gene B encodes for enzyme B. And these enzyme A and B works on the precursor in the similar manner and produces the same type of the product. So the alleles of both the genes expresses the same phenotype and it is triangular, and these do not show the cumulative effect when present together. So all these genotypes give the same type of the seed capsule, that is triangular shaped. In this genotype, alleles of both the genes are in the homozygous recessive condition and seed capsule is top shaped. Number is 1. Here, in the absence of the dominant allele A and dominant allele B, neither enzyme A is present nor enzyme B is present, hence it doesn't function on the precursor and forms the product. So it gives the different type of the phenotype, that is top shaped. In this way we get 15:1 ratio, and this is the phenotypic ratio. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.